Welcome, everyone. All right, so this session is I Have a Gut Feeling. You guys know the presentation disclaimer because it's been on every session that you've been to so far. This will be chaired by myself and my co-moderator here, Dr. Darren Patel. Here's some relationships for me that have nothing to do with what we're talking about today. Same for him. So really our objectives of this session is to really kind of appreciate the physiology, the variability of the intestinal pH along the GI tract and understand how the CF, TR protein impacts the pH and motility, compare these different options for screening for colon cancer, and then really look at the relationship of the CF metabolome and GI endpoints following ETI therapy. So here are our speakers, and it's kind of broken down into 3 sessions. The first 2 speakers are going to be Dr. Sellers and Dr. Davies. And what I really think is interesting is we've gotten really interested in this world of patient-reported outcome measures, or PROMs. And so in North America, or at least in the US, myself and a few of my colleagues, we developed the Galaxy study and the questionnaires that were going along with it. And simultaneously in Europe, you had the CF abdomen and the iterations of that. And one of the things that I found really interesting is when you look at the research in the US North America studies, we're largely showing that ETI doesn't have a huge impact on GI symptoms, at least not statistically, while in Europe we're seeing a little bit different using different outcome measures. So why are we seeing those slight differences? And maybe we'll get into that a little bit today. Then we have Mimi going to talk to us a little bit more about the metabolome and CF. This is obviously a topic of great interest across organ groups, and it's kind of hard to have a GI session these days without touching the metabolome at least a little bit. And then because this is a GI session, we got to look at endoscopy stuff, right? So Jake Robson and Christine Hetcham are going to talk to us about some different endoscopy things. And I think maybe, no offense, Jacob, but I think the interest in colon cancer screening and everything is on the forefront of a lot of people's minds. So we're really interested to see what Christine has as an update there. We will do questions and answers after each session. And you're already tired of hearing me talk, so let's get it started. All right, it's my pleasure to introduce Zach Sellers, who has dual appointments now as a scientific officer for 4D Molecular Therapeutics and remains an adjunct professor at Stanford University. And Zach, you should be ready to go. Here we are. Thanks, sir.
All right. Thank you, Jay, and thank you, Darren, for giving me the opportunity to share some data on this, and it's great to see everyone here this session. So I'm going to talk about the potential impact of elexacaftor, tezacaftor, ivacaftor on gastrointestinal pH and motility in CF adults, and these are results from PROMISE study. As is indicated here, I didn't collect all this data. I was part of the analysis team, and so it's really on behalf of all these individuals, and thank you to everyone who's involved in PROMISE to make this study happen. Do have some disclosures, but they're not related to this talk. So for background, I think all of us are very well aware that ETI has been shown many times over to improve sweat chloride, pulmonary health, whether that's by FEV1 or pulmonary exacerbations or other metrics, and also BMI. However, as Jay mentioned, I think the potential impact on ETI and gastrointestinal health has been mixed. There's been variable results on GI symptom relief, as he mentioned, from PROs. Studies have shown no change in stool frequency or consistency, although there have been statistically significant changes in improvements in fecal calprotectin. Many people have looked at the microbiome. We're going to talk about it some more today. And from some studies, we see that there may be statistically significant changes that occur, but sometimes those changes are probably not really clinically meaningful, and we put them in the context of what sort of a quote-unquote normal microbiome or a microbiome from a non-CF individual might look like. So we remain, I think, very confused as to what's happening in the GI tract, especially with ETI and even other modulators. So PROMISE study has been talked about at other meetings, and I think most of us are aware, but this is really a post-approval, real-world observational study to understand the effects of ETI. There are multiple sub-studies that were done, but within that, there was a pre and post ETI wireless motility capsule. We use smart pill, referred to as WMC here, that were performed in a subset of subjects, and it's that data that I'm going to talk about today. Now, wireless motility capsule allows for the measurement of intraluminal pH, pressure, and temperature in a non-invasive way. It's been previously shown that WMC can be used to identify decreased intraluminal pH, both in the proximal duodenum and the distal colon of adults with CF. So here I'm showing data on the left from the proximal duodenum that was first shown by Daniel Gelfand in the blue boxes looking at non-CF controls, and in the orange CF individuals looking at the intraluminal pH. And I think you can appreciate pretty easily that the CF individuals had a decreased pH in this segment within the proximal intestine that took a lot longer time to sort of get to a more near neutral pH. We also published using that same data set, looking at the distal colon, and found that there was decrease in the pH in the distal colon using the same data set and metrics. So the goals for this study were to analyze the capsule data obtained from PROMISE to, one, see if ETI treatment alters luminal pH. And we wanted to do that not just in discrete segments like the proximal duodenum or the distal colon, but really look across the entire GI tract. And then we also looked at intestinal contractions and motility indexes using the same data set. So here's some information on our study design. So there were 15 CF adults within PROMISE that were enrolled in this sub-study. For healthy controls, we used data from a prior capsule study, that one I mentioned from Daniel Gelfand. The design was similar to the prior capsule studies, that participants couldn't have any gastric acid suppressants for at least 1 week prior to WMC testing. And these subjects were on ETI for about 6 to 12 months. It was actually longer than initially anticipated, but because of COVID and our ability to do these repeat testing, it actually ended up being on longer than originally planned. But I'm not sure that's a bad thing. I think it actually probably helps us a little bit. Here are the variables we analyzed. I won't read through all of these, but usual demographics. And as I mentioned from the WMC, we looked at regional pH, pressure. We used temperature to help define some landmarks. And then contractions are defined based on the location as indicated, and the motility index equation is there should you want to recalculate anything. Okay, just a little bit about what this data looks like for those that may not be familiar with it. As I said, we extracted data from the entire GI tract using the GIM software, and we use sort of natural physiologic changes that we see as sort of landmarks. And let me see, can you see this? Oh, okay, great. So we use temperature to define when the capsule goes in, and then when it leaves. And then this line here, so you can see it's in the stomach, it has a low pH. When the pH rises rapidly, you know it's gone from the stomach into the proximal small intestine. Naturally, at the ileocecal junction, as it goes from the distal small intestine into the colon, there's a drop in pH. And then again, you know when it leaves because of the temperature change. So those help define sort of big categories for us. And then we broke that up into smaller categories, and we wanted to look 1 hour on each side of those category places that we could define. So we defined about an hour in the stomach, the proximal duodenum, the distal ileum, looked for a midpoint for the mid-small bowel, and similarly for the proximal colon, we knew that where that was, and the rectosigmoid colon, and then sort of took an approximation where we thought the mid-colon might be. So this is the data I'll use. Here are the statistics, mean standard deviation. We did paired Wilcoxon matched pair sign rank test for pre and post comparisons. NOVA, significance was determined using standard p less than 0.05. We set a priori sort of a trend mark that we want to see where there's significant trends in this, because it's not a big cohort. So that was predefined as indicated. Okay, so results first on our cohort. So there were 13 of 15 individuals completed both pre and post study that had usable data for us. Here's their information. As you can see, most of the individuals are F508 delta homozygous, and most are on prior CFTR modulators. I will also talk about some other subgroups that we looked at, and here are their characteristics. So we wanted to look at those who had not been on a prior modulator versus had, and also those that were on F508 del homozygous versus had F508 minimal function, to see if there were any differences in the response there. Of course, we're starting out with a relatively small cohort, and more times you start dividing this cohort, it gets smaller and smaller, so you have to keep that in mind as we go through the results. So first, I want to walk you through the intestinal pH results. As you can see on the y-axis, we have the mean intestinal pH for that 1 hour versus the different regions that we have on the x-axis. The red circles are pre-ETI, the white or open circles are post-ETI, and then in the black circles are healthy controls that we had used to give some context as to what changes or not we may be seeing. So when we look at statistical changes across in the entire regions, we don't find any significant changes with ETI in the intraluminal pH. We see in the mid-small bowel and distal ileum, in our sort of predefined trend regions, maybe there's some trend towards a change, but nothing significantly different. So we started to ask ourselves why that might be. When we looked at the cohort that had not been on a prior modulator, sort of hypothesizing that, well, maybe there had already been some benefits, that we're not seeing changes here. This is within this group, within the mid-small bowel, we actually can see a statistically significant change in pH. Now, it's actually a decrease in the pH change rather than an increase, as we typically are thinking about, oh, is there a decrease in this pH because of lack of bicarbonate secretion and other things. But when you put it in the context of the healthy controls, which actually are lower, have lower pH than the pre-ETI CF, it's sort of trending more towards that normal situation. There were no significant differences or trends at any of the locations when we looked at those that were only F508 del homozygous variants. We then wanted to look at the motility. We first looked at contraction frequency, and here the only difference in how the graphs look is instead of red being pre-ETI, blue is now pre-ETI, and those changes. And again, you can see is there's no significant change in contraction frequency with ETI. There's a trend towards a decrease in the contraction frequency in the proximal colon. But what you can see is compared to the controls, it's actually going in the opposite direction of the control cohort. When we looked at the Camilleri index, again, for most of the GI tract, there were no changes with ETI, although now when we looked in the proximal colon in that same region from contraction frequency that was trending towards significance, we did see a statistically significant decrease in the motility index in that proximal colon. But again, it's in the opposite direction of where our sort of healthy controls are. We found similar results when we analyzed those in individuals that had no prior modulator use or those that were F508 del homozygous. They both showed either a trend or significance in that same region in the proximal colon. So let me summarize what we've seen so far, and maybe we can sort of talk a little bit about now, and then even sort of maybe some questions about why are we seeing what we're seeing, because it necessarily wasn't what we thought maybe we might see. So generally we saw that ETI did not cause significant changes in intestinal pH in this relatively small cohort. We didn't see any decreased pH changes that were a baseline that had previously been observed in the G551D study. So let me just go back to this, maybe easier to see. I didn't spend a lot of time on it, but when you look at, so there's no changes here, but when you look at our pre-ETI values, let's just focus on the proximal duodenum, right, because that's what's been shown with ivacaftor to see a change. We see that our pre-ETI values are very similar to our healthy controls, right? So I think this makes me wonder a couple of things. One, are these the right controls to use for the study? These controls were used from a study decades sort of ago and were designed for that study. So I think it's worth sort of considering, expanding our control cohort to make sure that we can really validate our controls. But I will say that we have used those same controls and looked at other data like lumacaftor, ivacaftor data, and the pre-ETI values are different than controls there. So I think maybe less so. I think the other potential is that even though we looked at those that had been on no prior CFTR modulator and really didn't see much difference, it's a very small cohort. And I have to wonder if for all these individuals that were on prior CFTR modulators, had they seen some change already, which would mean that their pre-ETI levels are pretty similar to controls. And so they're really not getting any added benefit with ETI for this particular metric on intraluminal pH. We saw that ETI appeared to decrease contraction metrics in the proximal colon, but not elsewhere. And again, this was sort of in the opposite direction of our non-CF controls, not reason becoming more normal, if you will. I think we could think about the same control caveats as mentioned above. But again, we've seen actually similar directions in this when we looked at some of the other CFTR modulator data that I'm not talking about. So I actually think it's more complex than that. I think that what we'd need to do is probably see what is really happening at a contraction phase and look at this in more detail. So what could we do moving forward? So as I mentioned, we've been analyzing some of the data from GOAL and PROSPECT, really looking at the entire GI tract and to put the ETI data in context, and that can give us insights. I think we should make sure to look at some other controls to really validate that our controls can be used across all three of these studies that are done over very different time points. One can start to design experiments using that, for example, patient-derived organoids to do in vitro studies looking at acid-base transport with different modulators and see if there are differences with ETI versus other ones. And then for the contraction, I think likewise we can do in vitro contraction assays in the lab to really understand the contraction dynamics that occur with and without modulators. We can also do in vivo motility studies, really, and I'll defer to Doreen on this as the motility expert, but really proper motility studies using sort of catheters to understand what is happening with pre and post these drugs. So with that, I'll end and answer any questions.
Dr. Wolshansky.
Great talk. Thanks very much. Hopefully I'm not taking away from Jane's talk, but reflux improved though in these patients though. Right. So there is some sort of, there must have been some sort of change in acidity either in the esophagus rather than in the intestine. If you can explain that.
Yeah, so I mean, I'll comment really here is on the intraluminal pH as it's measured by the capsule, right? I mean, obviously, real life and, our GI, physiology can't be fully captured just by the capsule, itself. We've seen a number of studies, pre-modulators and about with variable, I guess, results on reflux or not reflux, increased risk, not increased risk. There's a lot of variability, as we know, in our patients. I think that there's always some of the challenges that we have from, this is all the data that we have on these capsules, right? I would love to have 10 times this amount of data, right? So that's always part of an issue. So I think there's multiple reasons to do that. The stomach pH, again, can be variable. We actually find sometimes more alkaline stomach pH and CF than lower pH.
Rosera Bass, Nationwide Children's Hospital. I was also interested in the stomach pH, and particularly how many of the subjects were on PPI, and do you feel like the 1 week discontinuation before the study potentially changed whether people were having rebound acid secretion at the time of the study in the stomach and small bowel?
Yeah, so I just put this up there because we're trying, so we can all remember what the actual data sort of is. So sort of pre-ETI, and there's no, I don't think it was statistically different than the healthy controls, but it's not less, certainly, right? So as to, is 1 week really enough to do it? I wasn't involved in the original design of the study. So the data was what we had to do with it. I think certainly we know that at patient levels, they have different responses to how they come off. I also can't tell you, I mean, Daniel can probably tell you, but how they sort of stopped off, if they were tapered off, but they were off for at least 1 week.
But if we were to do this prospectively, it would have been a little bit more time than 1 week. I agree with that.
I think you could.
So related to that, Linda Cummings online asked if they were on stimulant laxatives or any other medications that could impact motility.
Wireless motility capsules are not the best test for motility in the clinical realms. It's actually a solid-state manometry catheter, which is invasive, and we can't really do it prospectively as of now unless you have clinical indication. So it is possible that that may have affected the motility, because if you pull up the equation on the index, it relies on the sum of contractions and the number of contractions and amplitude. So if you have capsule stuck in the part of intestine where it just goes back and forth, because it's not an easy passage, sometimes it goes back and forth. And if you're on stimulant medication, it may have erroneously given us higher index than normal. Hard to comment, but these patients were not intended to study the motility. Remember, we went back and looked at the motility, but it was supposed to be more of a pH test and analysis, which there is no reason to stop laxative, not Miralax, not stimulant laxative. So it could have affected, like I said earlier, if we need to design this, probably given 72 hours of no laxative and then do the study, but that was not the design.
Let's try to get through these 3 questions in the next couple minutes so we can.
Okay, I'll do my best.
Thanks.
This is slightly a 2-part question. So the first question I have is, do you guys know how the mucus layer in the gut has changed after ETI?
We can't study that with this. I mean, there are people looking at that both at in vitro levels, primarily at in vitro levels, I'll tell you. But yeah, this doesn't pick that up. And so you do have, again, the capsule is measuring it as it sort of goes through the lumen, it's bouncing around, and so it may pick up things as it touches the wall or samples that, but those are probably brief. Yeah.
I'd imagine if that mucus layer is still there, that might, one, affect motility and pH level, because my understanding from the PROMISE study also within the microbiome realm that we haven't seen a difference after ETI in the gut microbiome. So part of me kind of thinks, and we haven't seen a huge change in like the lipids from the pancreas and stuff, that I wouldn't entirely expect to see a pH change if our biome is the same and everything else. But I do wonder if we were able to kind of change that mucus layer or be able to kind of get through it, that like those age of just having it there, if then we would see the change in the biome, but then also then see the change in the pH level. So that's kind of, I was curious about the mucus layer, if we have any ideas with that.
Yeah, I mean, I think those are all good thoughts, and I think, again, in vitro, we can probably get some answers to that a little bit. I agree with all the things that you said at the beginning about how these things may impact. Again, we have seen, not showing the data here, but we have seen differences, like, for example, with tezacaftor or lumacaftor, ivacaftor. Again, how these subjects are sort of changed, since being on that, and we just don't know. But I think these are all good questions that we need to be thinking about, right? And saying, we didn't see what we thought we might see.
Yeah.
Why?
Yeah.
Right. And so hopefully it's very stimulating to sort of dig into this deeper.
Yeah. Okay. Thank you.
Yeah. Nadia.
I mean, Yale. Zach, congratulations. These are very, very important studies to do. Quick question about, is there any data in vitro in intestinal cells expressing CFTR, looking at the effect of ETI on pH in the lumen or in the culture system?
So I can tell you, when I had my lab, we were doing these, and it was focused on duodenal enteroids and measuring chloride secretion and bicarbonate secretion. And looking at, we didn't look at across all of CFT modulators, but we did look at ETI sort of effects. We're still analyzing and getting that data out. But what I will tell you is we could see some changes, for example, and we looked at both crypt and villus enterocytes and things. We could see that ETI could increase cryptal chloride secretion, short circuit current, but it didn't necessarily change pH. And even when we looked at sort of villus enterocytes, where we think maybe that's more of the anion exchange is sort of happening, we didn't sort of see those changes. Now, part of the challenge, as you know, is in these in vitro systems is we're piecing together different populations of cells, studying them separately. And what we really need to do when we have tissue is all these processes occur together and they interact with each other, right? And so I think for those in vitro assays, they need to become a little bit more complex for us to fully understand. But I do think from what we were able to see in vitro is that ETI may be doing different things, whether it's versus chloride versus bicarbonate secretion, and that may be on the cell types that they're impacting.
It may be worthwhile to do some studies in vivo with an animal model to look at the effect of the ETIs on the pH.
Yeah.
Yep. All right.
Yeah.
So let's take that a little bit offside. Let's let Dr. Borowitz give us a comment because she did a lot of this sentinel work.
Yeah, I did. Drew C. Borowitz from Buffalo. I was part of this original work. And so I think this question is why? Why this is surprising results. So it could be maybe that the original results with ivacaftor were wrong, except the effect size was so large. So even in a small population, we saw these changes. The capsule has not changed, so it could be that the detection method is wrong, or it could be there's something about ETI. So the potentiator, ivacaftor, seems to have an effect on bicarbonate, but it's combined here with two correctors. And so one possible hypothesis is that maybe there's some interaction there that enhances the chloride transport of CFTR that's been rescued with ETI, but maybe interferes with the bicarbonate transport. And I think your in vitro work is really important in that way. And we need to sort of think about this. We have this great test for chloride transport, but we don't really have a good way to look at bicarbonate transport. So thanks for presenting negative results, which are as important as positive.
And just everybody know that, capsule or smart pill has been discontinued as of now. So they were not getting more capsules or support from company. So this is all retrospective. So if we need something in this realm, we need to really have need to wait for something new that's upcoming. They have not announced it, but I have heard, but unsure, or do the solid state manometry, which is more invasive. Thank you again for this wonderful talk. With this, we will move on to our next speaker, Dr. Jane Davis, who is going to talk about improvement in abdominal symptoms, CF ABT score, and gut inflammation over a year worth of treatment with ETI in children from 6 to 11, result from RECOVER study. She's from Imperial College London.
Thank you very much, everyone. I feel a little fraudulent. I don't normally come to these GI sessions, and I've never spoken at one before. I'm replacing Paul McNally, who's been given another talk in a different session at the same time. So bear with me if you are going to ask difficult GI questions at the end. So the RECOVER study is an observational long-term study which is co-led by Paul McNally from Dublin and myself from London in the UK. It's a multi-centre study looking at multiple different domains that change when people clinically get prescribed onto ETI. The lead of the GI arm of this is Jochen Mainz from Germany, and he's not able to be at the conference. So we're looking at multiple different domains, and I'll talk today about these GI symptoms. Here are my disclosures. None of the research grants specifically for this project came from Vertex. So RECOVER, as I said, is a real-world study of ETI in people with CF. We have 2 separate cohorts based on the timing of reimbursement in the UK and Ireland. And so we first commenced with children and adults who were 12 years and above, and then later on recruited a pediatric age group between 6 and 11. So as I said, this is a multi-centre study. On the left there is our centre in London, and then in the Republic of Ireland and Northern Ireland, there are multiple centres which are taking part there. We've got a lot of outcome measures, and you may have seen Paul or colleagues presenting on others of these, either previous conferences or now. But those that I'm going to focus on today are the abdominal symptom score, which was created by Jochen Mainz, and fecal calprotectin and N2PK, and fecal elastase. So first of all, a little about the score that was devised by Jochen and his team. This is called the CF-ABD score. It's a specific patient-reported outcome measures for cystic fibrosis, and it comprises 28 questions in 5 different domains. And you can see here the first publication in 2017 of a relationship between ultrasound findings and symptoms obtained with this score. We've used this previously and reported in the RECOVER cohort that was older, that was 12 years and above, and noticed significant improvement in this score at 1 year. And this is the data that was alluded to earlier, which seemed to be discrepant with the symptom scores that are coming out of PROMISE. So you can see here how this works. Here are the 5 different domains around the side of this spider plot. So pain, gastroesophageal reflux symptoms, bowel movements, disorders of appetite, and a quality of life impairment. And then these are combined into a total score, which you can see at the top. So these data have already been published. What you can see on the spider plot is before ETI in red, and then a shrinking of these areas after 2 months, which is in green, and after 12 months, which is in purple. And these were significant in all of those domains. Also in that cohort who were 12 years and above, we took the opportunity to look at fecal markers of disease and their improvement. And you can see here a measure of inflammation, fecal calprotectin, in 73 people who had 3 paired data sets, baseline 1 month and 6 months, and significant decrease in that inflammatory marker over time. Also a significant decrease in the M2PK, which I understand is thought to be a measure of cell turnover, and a significant increase in fecal elastase, although in this older aged group, not above the cutoff that would normally mean anybody would be able to come off their pancreatic enzyme treatment. So that was just to set the scene for what we'd seen previously. I'm now going to come on to the data from the current cohort, which is 6 to 11 years of age. You can see reasonably good numbers here across this table in terms of stool samples and CF-ABD scores at baseline, and then repeated measures for the first 12 months. We don't yet have the 24-month data, which is awaited. This was analysed statistically with a linear mixed effects model for repeated measures to test for difference from pre and post, and to also look for various variables, including sex, other modulating therapies, and genotype. I'm just going to show you the group data today. So here we have the abdominal symptom scores. From the top left, total score, and then all of the domains that I just highlighted on the spider plot. And this is the time points over that 12-month time period. And you can see for the vast majority of these, certainly 5 out of the 6, there is improvement statistically at least one of the time points. And for the total abdominal score, there's significant improvement at all of those time points. The quality of life was the only one that didn't show significant. You can see there is something that one might call a trend there. So how did the total abdominal score changes compare with the older age group? You can see that here there's very, very close relationship between this younger age group and what we'd seen already in the older age group. Things did differ a little, though, when we come to look at the biomarkers in the stool. So these are slightly busy graphs, but what I've got here in the lighter turquoise is the younger age group, so the area of focus for today's talk, and then the darker turquoise is the older age group. And you can see, I think the first thing to see is that in terms of fecal calprotectin, the baseline values are not as abnormal in the children as they were in the adults. There is a statistically significant, albeit small, reduction at 1 month, and then the change at 6 months is no longer significant. So that differs quite dramatically from what we saw in the older age group, where there were significant differences at both of those time periods. And similarly, the percentage of people with M2PK levels above a cutoff of 4 units per mil, that probably makes a lot more sense to you than it might to me. We saw that was significantly different in the adults in the darker turquoise, but that was not different in the children. There was overall a much smaller percentage of the children who had that abnormality, and that didn't change over time. Interestingly, the only one that I expected might be more impressive in the children than it had been in the adults, really didn't show very much at all. So we can see here, if we look first of all at the dark blue, the adult data, here's that significance that I demonstrated earlier. In the pediatric age group, the values were actually much higher at baseline, and they stayed pretty static throughout the duration of the study. That said, there were a few of the pediatric cohort who had values above the 200 unit cutoff. That proportion didn't increase. This slightly surprises me because it differs to what we see in the clinic and what is reported anecdotally, and certainly what we see in the slightly younger children below the age of 6. But certainly the numbers, I think, are pretty big and robust, so I think the data have to be taken as they are. So in conclusion, the improvement in abdominal symptoms with ETI is sustained for up to 1 year in children with cystic fibrosis aged 6 to 11 using this CF abdominal score. However, the markers of gut inflammation, cell turnover, and pancreatic function were less abnormal in that pediatric cohort than they had been in the adolescents and adults, and they either don't improve or don't improve as markedly on ETI. In terms of future directions, we will be completing the data analysis for the whole of the 2 years in both of these cohorts, and at that time point, we'll drill down more into the different subgroups based on gender and genotype, for example. Microbiome analysis is ongoing, and we will integrate those findings. The purpose of Recover having so many different domains is so we really drill down and interrogate relationships between them and understand mechanisms as well as just presenting what we observe. And we've also had funding and approval to do a longer-term extension of Recover, which was initially designed and funded to be a 2-year period. So we've actually got people continuing to have these measurements done, albeit slightly less frequent time points as time goes on. We also have 2 new studies, Enhance and a sub-study of that called Bunny, which are seeking to understand the evolution of cystic fibrosis disease progression from birth onwards. So Recover only recruited from 6 and above. We're now recruiting babies from newborns, and we're actually looking at a number of these markers in a much more light touch study. So really at the times when these babies and children are coming to clinic anyway. But I hope that we will get a bigger understanding of the progression of CF in the current era. And of course, many of those children in early age will be going on to ETI, and we'll be able to follow some of these markers there. So I would like to thank everyone that's been involved, in particular the funders of Recover, which was largely the CF Foundation, but also the UK Cystic Fibrosis Trust and CF Ireland. And then you can see the very large number of people who've contributed to this data set, many of them working very hard in the background. So thank you to anybody who's here for all the work. And thank you very much.
Wonderful talk. First of all, I'd like to bestow upon you the title of honorary gastroenterologist. I think it's wonderful. And I'll send all my patients with abdominal pain to you now. It's really... Jane, I think that at one point, I just want to say that I think if you start off with the normal calprotectin, it's not going to get more normal. So I'm not worried about that at all, about that particular part of it. That's all I wanted to say, because we shouldn't be despondent about it. The elastase, I don't know, maybe in longer term or with younger children, but I think the results are very, very important, even though they're not positive. Thanks very much.
I hope you don't think I'm despondent about any of this. I'm delighted if we're starting these drugs before anything's become abnormal that we don't see any change.
Good, Zach.
Thank you, Michael.
I've two questions for you, really about the abdominal score and the results that you have from there. So the first one is what I noticed when you look at the time dynamics, that you see pretty rapid sort of decrease in most of them at 1 and 2 months, and then you seem to see a sort of a rebound effect, sometimes even back to baseline levels at 6 and 12 months. So my question is, can you hypothesize or comment about why you think that may be happening? Is it physiologic changes? Is it people just changing how they're feeling and reporting out? Any changes in subsequent medications and like that? And then I guess the second part, what may feed into that, is all of these individuals know they're on the drug, and they're sort of looking at, do you have any data even from other studies as to how well this score performs and with sort of a placebo sort of arm that you can sort of extrapolate as to what effects could be related to that?
So really, really good questions. I wish Jochen were here. I don't know of placebo-controlled data in which he's road tested this score. I suspect people in the room would know if they had been published. I don't think so. No, so that is, of course, a limitation, and you're quite right. There's a big connection between mind and gut, isn't there? And and I do wonder if part of that might relate to this sort of potential plateauing and somewhat of a rebound. People have described to me their feelings of immense relief when they start to lose a symptom that they've lived with for many, many years. And maybe that relief then gets tempered over time because it becomes the new normal. I really don't know. We certainly don't have data to say whether this is clearly explained on a physiological basis. But I think what we will be able to do ultimately, when we have all the time points from both sets of patients, is have a look at relationships, if they exist, between, for example, changes in the CF symptom score, sweat chloride, some of the inflammatory markers in the gut, etc., and see whether there's anything we can tease out. And I think that's going to be almost doubly interesting than just reporting the findings, I think.
Absolutely. Great. Thank you.
So just a little bit to add. So I think part of the CF abdomen score, what's great about it is CF specific and it's validated in CF. It also, though, measures over a pretty prolonged timeline. Now, he's done a better job of actually cutting that down, and they still haven't gotten to the point where they have an MCID or a medium clinical important difference yet. So if you look at a lot of these studies, they start with pretty low symptomology, and they statistically have less symptomology, right? And so there doesn't seem to be a huge change from that standpoint from a clinical significance. I think what's going to be really interesting is if you do look at those patients that have severe symptoms and see how those scores change, that might be clinically impactful, which does mirror a little bit of what we've done with the Galaxy study as well.
Mike Narquist, great GI talk. It's more a plea, as you're one of the leaders. This study is very similar to PEDS Promise, which enrolled the exact same group, which do have some differences. They have higher fecal calprotectin in those, dramatically improved, look like the rest of Promise, but have those. I think one of the things that we should do internationally is combine these data sets where we have similar outcome measures and really look for those tighter associations, like people with high symptoms. Is their microbiome very different? You're going to struggle with numbers just like we're going to struggle. So that's sort of the plea. And Paul and I have talked about it.
So very much after my own heart. And in fact, we have been talking quite closely to some of the Promise investigators and had our first joint output at one of the conferences last year, which combined the entire sweat chloride data and added an enormous amount of power in terms of numbers. So we definitely would like to do that for multiple other domains. And I know Paul and Jochen maybe have already been talking to you about that. So please do keep it going. Very grateful.
Hi, I'm Sarah Jane Schwarzenberg from Minnesota. And I really enjoyed your talk. I would very much like to investigate the differences between CF abdomen and the PAGI-Sim, PAC-Sim that we're using and try to understand why we apparently, I'm not sure it's really true, why we apparently get different results. I will also add that when we analyzed our early data, particularly at 1 month, and we did a lot of sub-studies. We looked at girls and boys and the PROMISE study and all of that thing, there were some changes, particularly in women, that have disappeared now as we move into the later months. And I do wonder about how much we saw sort of an excited early result versus a different later result as people live with it. But I think examining these things together would be really helpful.
Thank you. I agree. One of the other things that probably people didn't have time to see on the screen is that we're really interested in medication uptake. Now, we know that's not an absolute perfect measure of adherence, but I think there is a possibility that some of the flattening out or even rebounding we see in some of our markers is people feeling better and stopping taking some of their other drugs. I've certainly had quite a big cohort of female adolescents in my own clinic who stop their Creon because they are gaining weight and they don't want to gain weight. And we've got people who, of course, dropping their respiratory treatments. So part of it could be that, and we really need to drill into that if we can.
We'll have one more question.
Real quick, Jill Sullivan from the University of Vermont. In that medication adherence, have you looked at sweat chlorides as an indirect marker of medication adherence?
We have got sweat chloride data at all of the time points we're looking. We haven't yet done the analysis of comparing medication adherence with the sweat chloride or fully analysing that, but we will certainly be doing.
And could that correlate with the elastase?
Possibly. I think what we're probably going to see is that adherence is maybe best to ETI and it drops off in some of the other medications that are a bit tedious and take long and people don't want to do them. But I'm predicting, we haven't yet properly drilled into that.
Thank you.
Thank you so much.
Thank you.
All right. Next, we'll have Mimi Savirda. Sorry if I mispronounced that. Associate Professor from National Jewish in Denver. Get this started for you.
I'm a pulmonologist. Okay, so no hard questions, please. And I am personally very interested in how we turn food into metabolic energy. It is a process that affects every single one of us. It's very personal to everyone. No disclosures. And the reason why I became interested in it is that after ETI, our patients acquired a tremendous metabolic capacity. And that translated to the ability of young women to carry pregnancies to term, to even become pregnant and then carry the pregnancies to term. The ability of patients to manage their pulmonary exacerbations without medical intervention and reduce the amount of hospitalizations. And in some patients, like to the far right, actual physical transformations where you don't even recognize the person that you're taking care of. So how did metabolism change to produce all these outcomes? And then on the flip side of that, why did it not change in some people? So questions that come up in our clinic and in the hospital every day is, why did some people gain weight and others still struggle to gain weight? Why do some patients still suffer from DOS? What's happening in the liver after ETI? Dr. Narkowitz is here, and I think that data is being presented at this conference about that. And what's happening with lipids after ETI, because we're seeing big changes in their cholesterol panels. So there's really no better platform to look at these questions than the PROMISE study, which has already been mentioned. Huge pre-post ETI study, 56 sites with an amazing biorepository that not only captured core study endpoints of FEV1, sweat chloride, weight, patient-related outcomes, but also got into the GI as well. And when they got into the GI study, for those of you that are here that designed it, this was incredibly useful that you parsed your biomarkers into functions. So intestinal inflammation, malabsorption, pancreatic function, liver function. The leadership at the TDN was incredibly gracious, and they allowed us to do a sub-study looking at 3 time points in PROMISE. So PROMISE went out 4.5 years, and we basically were able to look at samples in a cohort at baseline, so before ETI treatment, 6 months after treatment, and 18 months after treatment. And the question that we wanted to ask was, did metabolite changes help explain some of those metabolic transformations that I just showed you after ETI? So I'm going to actually structure this talk into three parts. First, I'm going to talk about the metabolome in general. How does it change pre and post promise? Next, I'm going to talk about what do those metabolites that change, what do they mean? How do they relate back to clinical context that patients care about? And then finally, is there a relationship between all of that and GI homeostasis? So part one. So PROMISE, basically all we did was a post-hoc observational study. It was really quite simple. We wanted an accessible compartment, but we wanted that compartment, which ended up being serum or plasma, to reflect absorbed nutrients in the gut, products of cellular metabolism, so glucose, fats, amino acid, products of cellular waste. And really, because I work in inflammation, what we were looking at is we hypothesized that those primary metabolite classes would reflect the reduced inflammation that you see after ETI. But it went a little bit off the track. Here's a little bit about the study. So we had a cohort of 115 patients, pre-ETI 6 months and 18 months. Generally, they were a young population. They were mostly all non-Hispanic whites. Their FEV1 was pretty well preserved at baseline, so 83% on average. Sweat chloride came down significantly. BMI went up significantly. So overall, a relatively healthy cohort. We were able to look at 142 metabolites, and this is actually the machine where we did the work doing HPLC mass spectrometry and looking at metabolites across 8 classes that are listed there. So after the metabolomic score was able to process, filter, normalize the data, then we could perform unbiased profiling of 142 serum metabolites using MetaboAnalyst software. The figure on the left is particularly useful because it combines t-tests as well as fold change so that you get the most significant metabolites related to both. You can see succinate far and away above everything else, but there's other classes that are represented as well. Amino acids, oxalocrotonate, which is in the indole tryptophan pathways, sphingolipids, and then heme metabolites. To the far right is another way of looking at significance. And what that is VIP scores using a discriminant analysis. And so then you could look at each individual metabolite and how does it discriminate between the pre and post Trikafta state. And actually, this is just looking at pre and 6 months after, which is where obviously you see the biggest differences. This is over 18 months. So in red is at time 0, and then green is 6 months, and blue is 18 months. And you can see that some metabolites go up, some metabolites go down. Some of them go up and continue to be significant from time 0, and some of them actually start to lose that significance. And when you look at it by class, what you see are that fatty acids in general and a group of fatty acids called acylcarnitines go up, heme metabolites go up, TACA cycle intermediates go up. Amino acids can go up or go down, and indole and tryptophan and sphingolipids tend to go down. One way that you can look at nutrients and how do the nutrients that we eat relate back to the metabolome is to look at nutrients that are essential. Essential nutrients are ones that we have to take in through our diet. We as humans cannot synthesize these. So when you look at essential amino acids, they really are not changed. This is at the pre and then 6 months after Trikafta time point, except for one, histidine, which went up. Also, we can do this with fatty acids. And so pre and post Trikafta, if you look at, so there are 2 fatty acids, linoleic and linolenic acid, that human hosts don't have the enzyme to make. So they have to come in through our diet. They are eventually the basis for the production of longer chain fatty acids, which are the omega-3, DHA and EPA, and the omega-6 fatty acids, like arachidonic acid. And so you can see there across the board, none of them change significantly in this cohort. And so the story is not so simple as, okay, you got an increase in nutrition and therefore you're absorbing these more and these nutrients will go up. Some of the metabolites are not only coming in through the diet, but they're actually reflecting cellular processes. And so that's true for succinate and citrate. Succinate and citrate are both metabolites in the TCA cycle that is happening in the mitochondria, and it's critical to the production of ATP. They were both significantly increased at 6 months. Something that was very curious and very surprising to us was something that was happening with the fatty acids. Now, fatty acids are converted into different molecules in order to go into the mitochondria. So medium chain and long chain fatty acids are esterified to carnitine and then bound to an acyl-CoA group. Then they go into the mitochondria using the carnitine shuttle, and at that point, they're used to produce they're used to go through fatty acid oxidation, which produces acetyl-CoA, and it also contributes electron equivalents into oxidative phosphorylation. Fatty acid oxidation is a very energy-dense way of making ATP. It produces more ATP than glycolysis does. Across the board, and it really, like when you're looking at the data, it just jumped out. Medium and long-chain fatty acids, just boom, one after another, all went up significantly. So what we see then in part one is that there appears to be some sort of fatty acid oxidation signature related to the metabolome. Okay, so what does that mean? And what do these metabolites mean when you think about the things that matter to us as clinicians and in clinical trials? And so I just listed 3 metabolites: succinate, aspartate, sphingosine phosphate there. And you can see that there's some that go up and some that go down. And so doing modeling, statistical modeling, can really help you figure out, is it a good thing or is it a bad thing when those things are changing? So not only did we do mixed models where we looked pre versus post and pre versus post ETI, but we also did mixed models modeling changes in the metabolite associated with changes in FEV1, changes in sweat chloride, changes in weight. We weren't sure that there would be really much of a relationship between those, considering how different they all are. Much to our surprise, they were highly significant in every one of those 3 endpoints from the core study, from the core promise study. And so you can see the p-values are highly significant, as are the multiple corrections. And what was very useful is the coefficient of correlation and the directionality, so that you could see the metabolites that went up with FEV1, metabolites that went down with FEV1. Probably the most surprising was that the greatest correlations and associations were seen between metabolites and sweat chloride up to the -15 p-values. We were not expecting that at all. And the directionality was maintained. So succinate, which goes up with FEV1, goes down as sweat chloride rises. So the directionality made sense for all the metabolites. This again happened again when we looked at weight. Again, significant p-values matching directionality for these metabolites, and a lot of repetition of the same metabolites across those 3 endpoints. So I just went through the core study endpoints. Now I'm going to go back to the role of the GI tract, because we can do that using the PROMISE data set. And so when we break down those endpoints and we go into the 4 individual evaluations, and some of this has been mentioned here already. I am going to go over some of the data from Dr. Schwarzenberg's papers. I'm very honored that she's here. So when you look at exocrine pancreatic function, there were really not any significant changes in PROMISE at 0 and 6 months. Intestinal inflammation, there was an improvement in fecal calprotectin. It was significant, but it was a small effect size. As we've all heard, there were some improvements in patient-reported outcomes, tending to be more in the patients who had more severe symptoms. The one that's the least well understood is non-pancreatic malabsorption, fecal bile salt loss. And so there are 2 markers there that are very important, and I'm going to go over them a little bit more for you and for me in order to kind of really get a good sense of what they do. So FGF19 represents reabsorption of bile acids at the terminal ileum, and then there's an enterohepatic circulation, a negative feedback inhibition, and then C4 represents hepatic bile synthesis. And so FGF19 is usually low in CF because it's not being, they're not being reabsorbed, and then C4 is usually high in CF. Okay, here's a pictorial to help break it down a little bit easier and to help get your mind around what else is going on in the environment around the bile acids. So in the liver, cholesterol is basically emulsified into bile acids, which undergo a primary conjugation in the liver. Then they go into the GI tract. In the small intestine, they'll undergo a secondary conjugation. What's in the small intestine? There's dysmotility, there's low pH, there's bacterial overgrowth, there's dysbiosis. Those are not normal secondary bile acids. Then the next step at the terminal ileum is that bile acids should be reabsorbed. Normally, 95% of total bile acids are reabsorbed and 5% go out into the feces. In CF, there's an up to 3-fold increase in fecal bile acid excretion in CF. If you look to the far right, that shows data from from the PROMISE trial that was the cohort that we used. So what happened with those biomarkers in the cohort that I'm presenting to you today? And you can see, as expected, FGF19 went up at 6 months and 18 months in the PROMISE cohort, and then C4 went down because there was better bile acid reabsorption and more bile acids did not need to be made. So then we did the same thing, right? So computationally, we looked at what are the associations of those metabolites with the specific GI endpoints from PROMISE. And what we found was very interesting. When we looked at intestinal inflammation, yeah, there were a few associations with metabolites. There were no associations with exocrine pancreatic function and metabolites. There were many associations with non-pancreatic malabsorption, specifically through C4 and FGF19 and the metabolites. When I was reading about this, because as a pulmonologist, I don't know anything about bile acids, one of my favorite papers actually mentioned that bile acids are more than intestinal soap. And I think the more that I have delved into this, the more I feel like this is the case. The thing that I want to point out in this picture is if you see cholesterol and the primary conjugation that goes on of bile acids in the liver, the next step is a conjugation in the small intestine where they undergo the secondary conjugation. And so for this process to occur, there is dependence on the host and the pathogens within the microbiome. And so that is a very delicate relationship. And then ultimately reabsorption occurs in the ileum by enterocytes along multiple receptors. So what I've just told you is that bile is a physiologic detergent. It is important in enterohepatic circulation, but it is also managing a lot of our total lipid absorption, the way we metabolize lipids. I'm almost done. And so I want to just go very quickly over some of these associations to show you a very curious thing. So if you look here, these are the correlations with C4, and these are the correlations with FGF19. There were many more, but I'm just showing you, whoops. No, almost done. No. I'm just showing you a few. So with C4, right, so C4 starts out high in CF. You can see that some of those metabolites that were actually so closely associated with our important endpoints, the primary endpoints of PROMISE, are repeated there again. But what's even more curious is that if you look at FGF19, which is so important for bile acid reabsorption, one after another, you see the acyl carnitines listed there as well. So if these are mitochondrial markers of fatty acid oxidation, beta oxidation in the mitochondria, there appears to be a relationship between uptake in the terminal ileum and this increased metabolic currency that we see after ETI. One thing that I did not know is that different organs use different substrates in order to produce energy. And so beta oxidation is the primary form of generating energy in kidneys, liver, muscle, at rest and low exercise, and in the heart. So in conclusion, the metabolite change before and after ETI, it's significant. And it doesn't just signify improvements in the gut, it signifies improvements in cellular function. Those metabolites are highly associated with very important endpoints in CF, in clinical trials, and in the actual patient experience. Those metabolites associate with normalization of bile acid homeostasis in the gut, which actually appears to be critical in whole body energy generation. Who knew that? And the CFTR rescue, as it's reflected in the circulating metabolome, reprograms metabolism, and it indicates more efficient energy generation through the contributions of fatty acid oxidation, which is now more robust, and better generation of ATP so that a patient can carry a baby to term or become a bodybuilder. This work was tremendously helped by multiple individuals. Many thanks to the folks at the CFF TDN, the PROMISE investigators, in particular Scott Sagel, who made it all possible, and our group, a tremendous group in our lab coordinating all these samples, and then the metabolomics core, who were tremendously helpful with the analysis. The computational part, those are fantastic computer scientists. I believe that these are the ways we need to be thinking about these big repositories of data and how to connect gut, lung, gut, brain, all these connections that we're seeing with the drug that affects so many parts of the body. Thank you.
So very interesting. While we wait for anybody else, I do have a question. So interestingly, I do TPIAT, so total pancreatectomy, islet autocell transplant. Okay. So take the whole pancreas out. And yet we've got a cohort of patients who, without having a pancreas, do not need pancreatic enzymes. They are somehow gaining weight, not having vitamin deficiencies, not having significant steatorrhea. I'm not going to say they have a completely normal diet, but they still have fat in their diet. And so it's gotten me thinking about what other intestinal adaptation may be occurring. CF specifically, we have these patients going down on their fecal elastases may or may not change. We have a love-hate relationship with fecal elastase, but they're definitely taking less enzymes and seem to be doing well. So that's a long way of asking, do you have any other signal here you think that shows some other non-liver, non-pancreatic malabsorption intestinal adaptation signal, or any way to look that through these metabolites?
So what I really love to do is get my hands on some more of the GI endpoints from PROMISE, because I had a very limited amount, so that I can actually try to parse together, at least statistically, where those associations and relationships are, so that we can start to go down those pathways.
Dr. Hoffman?
Dr. Hoffman.
I can actually, along those lines, this is not a question. This is in response to that question. I just want to point out that in the fecal samples, we are going to be doing some metabolomics as well, which would be fascinating to compare with to try to get to that question. Actually, Nicole Green in the back is probably going to be looking at the metabolites, so we can compare metabolites before and after. But that brings me to my question. Mm-hmm. How confident can you be, and are there any clinical data regarding diet from the PROMISE study? I actually don't know. How confident can you be that what you're seeing is the effect of ETI on either absorption or metabolism in the gut, especially, or in the liver, especially regarding bile acids, as opposed to a change in either nutrient intake, or diet, or either change in diet or just change in volume of nutrient food?
As confident as you can be with a post hoc observational study.
Are there dietary data? Do we have dietary? I don't even know.
Alex, do we have? No. Did you guys collect that?
He said no.
Oh yeah. Okay. Okay. Yeah, I don't remember that. I mean, I could say just in general, most of our patients did not radically change their diets after, unless they started gaining a lot of weight. But I don't know that.
Okay. Great talk, by the way. I
Thank
should have
you.
started.
Thank you.
Time for one more question online. Were patients fasted for or fed for the blood draws? And the second, in the same along the line, any data on changes in energy expenditure for those patients that could correlate with metabolite changes that are reported?
I don't know that energy expenditure is a part of the data set. Do you know? Yeah. And then Alex, were they fasting? Yeah, so that's useful. And then that does normalize it to some extent.
Right. Wonderful. That was a great talk. You do belong to GI community. All right. Our next speaker, like Dr. Freeman said, changing gear here. It's going to be Dr. Jacob Robson from Primary Children's University of Utah, Pediatric Gastroenterology, who's going to talk about endoscopy finding in a retrospective cohort.
Thank you for having me today. I want to make clear that I'm presenting today on behalf of one of our residents, Dr. Nate. We said, hey, why don't you look at all the endoscopies we've done over a very long period? And shockingly, he found the time to do this. Unfortunately, and not shockingly, he could not get the time off to be here because residency still is residency. And it's my privilege to present the work that we did together on his behalf. I want to say what a privilege it is just to be here. That talk made me really excited to be alive. It's so cool that we're looking at this right now. And it's so cool that we've really expanded our resources to do pediatric endoscopy. I'll talk about our volume in a minute. So the title of our talk today is Gastrointestinal Pathologies in Pediatric CF, Patients Undergoing Endoscopy, a Single Center Retrospective Review. I have no relevant disclosures to the content of this talk. So setting the stage, and this is probably shown hundreds of times at this conference every year, patients with CF are living longer and healthier lives. From the start of my medical school training, which was prior to newborn screening, we've had this explosion of coordinated care and medications, and we're watching patients, not just supporting them through their childhood, hoping they make it to a young adulthood. We're really encouraging and thinking about what their lives look like into adulthood. This was shocking to me when I looked it up a couple days ago, that we're beyond the 30s and 40s, and the median life expectancy is now in the 60s. With that, especially as pediatricians and transitioning care to adult doctors, we need to think about how to optimize health, not just survival, but health into adulthood for our patients. Certainly, we all are living in the era of CFTR modulators. We spoke last year about some of the ongoing disparities in care as far as who gets a modulator and modulator research, and I hope that will continue to be an ongoing topic of discussion. But just 5 years ago, we were looking at 50% of people who were either not eligible or not prescribed. And as we move into this year, we're looking at 20% and soon to be less. Many people now have access to modulators. To take you way back, when I started frequenting CF clinic as a medical student and as a resident, interested in gastroenterology, we were really focused on nutrition. We knew that bigger was better, in the sense that those that got to above the 50th percentile weight for length at age 2 seemed to enjoy better lung health, overall nutrition, and even improved transplant-free mortality into adulthood. So this figure on the right from Dr. Borowitz was sort of our Bible in clinic, trying to do everything we could towards optimized growth and nutrition, enzymes, consideration of acid blockers, counseling. We did a pretty good job. So even prior to modulators, nutrition was really heading in the right direction for children with CF. And I want to point out that this is sort of the era of multidisciplinary CF clinics. It's not just the docs, it's not just the medicines. It's the dietitians, feeding-focused psychologists, social workers, pharmacists, nurse coordinators, patient and parent advocates. I think our CF team dinner tonight is 50+ people from our humble small state, and it's because so many parents come and support their children, and many others. Also, I want to highlight the wonderful CF Digest Award, which many of us in this room have been able to benefit from, bringing gastroenterologists and really thinking about nutrition into CF clinic. The moderators had amazing premonition ordering the talks the way that they did today, because you've already heard a lot about symptoms. We talked that, nutrition was our goal. We were doing better with that. Now we really think about symptoms. And so in the Galaxy study, which I'll highlight first, this was a really nice way using tech and apps and everyone being tied to their phone every minute. Why not utilize that to see how our patients are really feeling on a day-to-day basis? And not surprisingly, those already on constipation meds, those already on acid suppression, a huge percentage of these patients are expressing really significant symptoms. So bloating, upper and lower abdominal pain, even vomiting. This is something that we need to do better at and is a great target for treatment. Particularly, this is both kids and adults. Up to 30+% of patients are complaining of straining with bowel movements or stomach fullness. I'll show briefly, the PROMISE study's already been really well highlighted today, but we're maybe even doing better with symptoms now in this era of modulators, although modest, as it is said in the abstract for this. ETI seems to be helping patients with their symptoms, and we're headed in the right direction here. Meds, multidisciplinary clinics, thoughtful care. So now that we're doing so well, what's left for us? Or now that we're doing better, what is left for me in the CF clinic? So to be honest, we often are talking about diet changes, intuitive eating, rather than just power packing. Maybe you don't want that fourth, helping of bacon and sour cream for the day. I talk about the low FODMAP diet in CF clinic more than I thought I ever would. Enzyme optimization. We're taking kids off PPI, not putting them on in kind of support of their microbiome. We're, still managing constipation aggressively. I will Miralax till I die. But we have these inklings, right? That patients are living longer with CF. We have 1% of the population with celiac. I'm going to talk about roughly 1% of our population in Utah having EOE. Why do patients with CF not get those conditions? They could, right? They probably do. So not shockingly, there's a lack of robust literature on this. But we were really interested, celiac, EOE, IBD, polyps, which I'll hold my breath on because I think that's going to be a lot of the next talk, or Barrett's esophagus. Why don't we know more about whether or not patients with CF get these conditions? So not shockingly again, I could find only a couple studies talking about endoscopy and results in patients with CF. So one was actually a capsule study from about 15 years ago. Most of the patients had abnormal visual findings and an elevated calprotectin. 15 were normal, so about a third were normal, 2/3 were abnormal. And then really one of the only pediatric study had 12 patients. 9 out of the 12, 3/4, had some kind of nonspecific duodenitis. It was interesting to me that 4 were classified as a nonspecific colitis with eosinophils, and then 0 were actually some other, clear mucosal disease on top of their CF. So we thought we would like to look at that. We thought we could move the needle maybe just a little. We have a lot of patients in our CF center, and so we thought we should look back at this and see and just present a little bit more data on this. So for those of you that don't know, the Intermountain West is a really interesting area for study. We are the only children's hospital in this fairly wide region. We're a hospital served by University of Utah physicians, but we also have the benefit of being under the umbrella of Intermountain Healthcare and have access to their resources. This allows for some population-based study and disease estimates. Very few people leave the center. We have about 1 million people in the region that depend on this hospital. and we have about 300 patients in our CF center. So with that, we reviewed 15 years of endoscopic data in those under 18. We had 72 endoscopy patients that underwent an endoscopy, all had an EGD, and 13 also had a colonoscopy. We looked at how old they were, their sex, their CFTR mutation, and whether or not they were on a modulator. Then at the time of endoscopy, we looked at their BMI, why they got a scope, and what the findings were. So here are our results. So notably, in our patient population, I thought it was interesting that, a few were pancreatic sufficient. It seemed like, a decent, that was an interesting finding that a few of them had an abnormal study, more so than in the normal group as far as endoscopy. And then very few were on a modulator. So that's a key limitation to this study, is that we really captured endoscopies before all of our kids were going on to ETI or probably a few on ivacaftor. So I highlight here that in the indication for endoscopy, we had a lot of nonspecific symptoms, right? Belly pain, poor growth, diarrhea, reflux, nausea. Those are all common complaints in the symptom trackers for CF. But a couple of these are quite specific. These are not nonspecific. When a kid in Utah says they had dysphagia, you got to think about EOE. Vomiting as well. Hematochezia, these are not things that I think of as common complaints within CF. So I thought it was interesting to try to pick out these indications for endoscopy. So here's the big money from this paper is that, so in the endoscopy, about 25% had results that we thought were distinct from their CF. So included in the normal pathology is when the pathologist says in the comment, like inspissated mucus or mild duodenitis consistent with CF. We felt that these 17 diagnoses were separate, kind of game treatment changing diagnoses for the patients. So I'll go back there just for a second and kind of highlight, as you can see. So we have some EOE, some celiac, and then a couple one-offs as far as more eosinophilic, or what we would call EGID, eosinophilic gastrointestinal disorder, or one patient with portal gastropathy. So let's talk about these results. So the majority of endoscopies were normal. This actually caused us to do a bit of a deep dive into pediatric endoscopy. So, well, that's a really high normal rate. Are we scoping too many patients? And it's actually fairly common in the pediatric literature to have a relatively low abnormal rate. A study from Dr. Thompson a few years ago showed that there's only about a 20% diagnostic yield, meaning a key treatment-changing diagnoses in about 1 in 5 pediatric patients that underwent an EGD. Certainly for us, EOE and EGID were most of the abnormalities, and celiac was notably less than EOE, even though people tend to think of that as a more common disease. So how many gastroenterologists in the room? Okay, a ton. So I'm not going to spend you've all heard of EOE, or many of you have. So the symptoms can be, I'll quickly say, more common in boys, mostly Caucasian. And the symptoms can be quite variable across the pediatric age spectrum. But as kids get to the teenage years, they tend to have more common what adults would complain of as dysphagia or food getting stuck. To get the diagnosis of EOE, you need to have symptoms, you need to get a scope, and then we need to kind of ensure that you don't have some other disease like a fungal infection. Just to highlight, in Utah, we are what I would call the world title belt holders of EOE. It's a separate talk about why we have so much EOE in Utah, but we have large families, we have some air pollution, we have good access to healthcare. Those are some leading hypotheses. So 1 in 1,000 kids in Utah were newly diagnosed with EOE over a 5-year span. Those kids are living into adulthood with this disease. We probably think that about 1 in 100 people in Utah have EOE. So that should be on our radar in our kids with CF. As far as celiac disease, everyone had an elevated TTG-IgA. We think this is a true diagnosis. That's pretty similar to some of the prior reported literature in CF with a, roughly 2% of the people having biopsy confirmed celiac that matched up with typically used serum biomarkers. I think one thing we have to stay tuned on, I'm expecting this talk in the coming years, is whether or not people with CF, what happens with the modulators. Do they, there's definitely a relationship between CFTR modulators and celiac disease. It's unclear how that will play out. There were some limitations, just that it's hard to get a true incidence and prevalence when people are coming in and out of the population. That's something we can look at over time. We're scoping a lot more. So we did, when I first started 10 years ago, we did about 1,500 endoscopies per year. Now we're doing 3,000. So I want to make sure we're being thoughtful about this. And very few of these patients were on modulators. I think if we look over this next 5 years, almost everyone will be on a modulator, and that will be really interesting. And certainly we're a single center with specific practices. I'm excited to track this over time to really be an epidemiology nerd with all of you and appropriately report incidence, prevalence. I think this is a great potential for a quality improvement project to standardize our practices around kids with GI symptoms and when to scope our patients with CF. We should probably scope more than we do. I think that we're probably missing mucosal disease that could be treated. And then we can pass these on to adult gastroenterologists, which I'm about to do right now, right? To talk about adult gastroenterology. Great. Okay. This would be a great role for CF Digest. This is in the world now. The journal Curious, if you haven't heard of it, published this paper, so you can check it out. This is Dr. Nate in the Uintas. This is me and my boys dragging them around Utah. It's a wonderful place to live. Come join us. That's it. Thank you.
Rosaire Bass from Nationwide. Did you look at post-procedural complications with endoscopy, and were those at all different in the CF population as compared to the general population?
Yeah, we did probably at all centers, the patients get a call a day or two after endoscopy that comes in. There were no significant procedural complications such as hospitalization or bleeding. So the answer is no. We didn't have any significant complications. Whether or not they had some mild belly pain, I don't know. We just looked, did anyone have to come back in or have a procedure? So the same, low complication rate.
Thank you for presenting that data. My question is, it's not uncommon to have increased eosinophils in tissues in CF. And since that's sort of the leading way we make this diagnosis of EOE, right?
Yeah.
My question for you is, do you think what you're seeing is what we typically think of as non-CF EOE that's occurring, so EOE in CF individuals, or is this more reflective of sort of the CF pathologies that there are just more eosinophils there and are not really the same etiological causes that we may see in non-CF individuals with EOE?
Yeah, great question. So I think the answer is yes for EOE. They have mucosal changes consistent with EOE, so furrows, plaques, loss of vascularity. So I think the eosinophils there are causing problems, and that tends to correlate with dysphagia. It's a great idea for a future study to really correlate the EREFs, the endoscopic reference score for EOE, with these findings. My anecdotal answer is that it's very high, and I think it's real EOE. For the EGIDs, for gastritis, enteritis, colitis, I don't necessarily think it's real. There's very few mucosal changes in those areas with these patients. So I think that the eosinophilia is probably more of a bystander in the lower GI tract, and it's true disease in the esophagus.
Thank you.
I think there is an online question there. What percentage of patients with EOE had symptoms of EOE?
Yeah, no, we didn't sub-classify. I did highlight that many of the indications for endoscopy in this population were dysphagia, which is a very specific complaint in EOE. If you want to get really granular, EOE is dependent on symptoms. It's a clinico-pathologic diagnosis. If a kid says they have belly pain and they do this motion for where it hurts, then technically that counts as EOE. I try to help them localize, like, is it in the top half? If it's down by your belly button, it's probably not EOE. But yeah, there certainly the dysphagia patients correlated with the EOE findings.
Hi, I'm Senthil Sankaraman from Cleveland. So a few years ago, we did a retrospect, like a couple of studies. So one is looking at the database to see whether there is increased prevalence. The answer is yes. About like, compared to general population, CF population has 6 to 7 times higher prevalence of EOE. And then also, so the reason why we looked into is in our center of like 100 kids, about like 4 or 5 kids had EOE, like classic presentation.
Mm-hmm.
And then it's always harder to see whether the symptoms are coming from CF or from EOE. That's always the challenge is there.
Agree.
And not just EOE, any mucosal disease you take, IBD, celiac, the prevalence has been like about at least 5 to 10 times higher, or like at least like 4 to 10 times higher in the CF population. A lot of theories are behind it, like dysbiosis, early exposure to antibiotics, PPI, things like that. So, and also the other reason is like CF population may be more accessible to medical care or interventions. And our threshold to scope these kids may be lower. So that may be another reason as well. Thanks.
Yeah, totally agree with those points. Thank you.
Okay, so I'm going to admit that I do not scope very often in this population. And you just told me I need to do it more, which I don't necessarily disagree with your data. But as you think through your own personal algorithm in your head, I'm sure with 75% being normal, it's not just belly pain, but what are the symptoms beyond dysphagia, which I think we would all scope for, that gets your attention, go, okay, this is the right patient that I need to look at?
Yeah, that's a great question. So one thing we're considering is making TTG-IgA a little bit more of our treatment algorithm. We've added GGT to our algorithms to screen. We're thinking about adding TTG-IgA in the right context for patients just with an increased celiac rate. You know, as far as EOE, kind of what I said previously, in addition to dysphagia, so we think about the male predominance, we think about if you ever are having animal protein get stuck in your throat, that is in Utah diagnostic of EOE. If kids can localize to the top half, if they say, I have chest pain, I have heartburn, I'm like my dad, I say, well, you're not supposed to have heartburn like your dad yet. So those kind of things, the upper GI symptoms make me more suspicious of EOE.
Time for one more question.
I was just wondering, so I have a patient and the patient has a lot of GI issues. I've never seen a kid with so many. And he's always being admitted and all that. Do you think that maybe it would also be more than just the CF?
Certainly could consider it. It depends on the specific symptoms, but yeah, no, I mean, I think that we're trying to open our minds to the fact that these kids are healthier and being treated so effectively that if they're still having specific complaints or issues, then it's worth taking a look.
Do you have any suggestions of like if we should look into it? I'm just a medical.
I'm going to interject real quick.
Let's talk.
The lawyers tell me we're not supposed to do that, but the two of you should definitely talk.
That sounds good.
Yeah.
Okay. Deal. Okay, thank you.
All right. This last speaker for the session, Dr. Christine Hashim. She's from where I am, my adult GI colleague at St. Louis University, and she's going to enlighten us about NICE-CF and non-invasive colon cancer screening. Dr. Hashim, please. All right.
Yours. Thank you. Sorry. Okay. I know I stand in between you guys and lunch, so thank you for staying. I'll be discussing understanding colorectal cancer and cystic fibrosis. These are my disclosures. I grant support from Exact Sciences, Polymedco, and the CF Foundation. So my objectives today are that we recognize that there is an increased risk of colon cancer and cystic fibrosis. I'll discuss briefly etiology and risk factors for colon cancer and CF, and then we'll discuss and compare potential options and screening. So as Jacob already presented, and this is actually a little bit older data, but it's still prevalent that most of our adult population is in CF is over the age of 18 now. And with improved survival, you can see the adult population is increasing. So 2019 registry data said median survival age 50 years. As you saw today in the symposium, it's increased to 60 years of age now. So with improvements in therapies, patients are living longer. And so that's all good news. This bad news of being an adult is that we have increased risk of cancers. And I will focus on GI cancers, but I will also highlight that we are seeing increased risk of skin, breast, and cervical cancer as well. So this is observed versus expected cancer risk in the CF Foundation registry. This is an analysis of U.S. cohort of over 40,000 CF patients. And you can see the registry data is from 1990 through 2009, so before modulators. And what I want to highlight is that on the left-hand side, you have non-transplanted patients, and the right-hand side, you have post-transplant patients. And the point of this study really highlighted that there is an elevated risk of GI cancers. So you can see GE junction, esophagogastric junction cancers, the absolute number is low, but the standardized incidence ratio is 3-fold higher risk. If you look at terminal ileal cancers, again, 11-fold increase. And today we'll highlight colon cancer, so 26 to 4 expected cases, 6-fold increase in non-transplanted patients, and a 30-fold increase in post-transplant CF patients. And why do we see colon cancer risk in CF? The CFTR is a tumor suppressor gene in the GI tract of mice. So when you have loss of CFTR function, you have GI tumor formation, leading to dysregulation of the immune response, intestinal stem cells, and growth signaling regulators. We all know that in our CF population, we have stagnant mucus, which can lead to bacterial overgrowth and dysregulation in gene expression, and epithelial homeostasis and inflammation. Hopefully, CFTR modulators can have an impact in this regard. What are some risk factors for CF-related colon cancer? Well, just like the general population, men are at higher risk. In our CF population, the severe functional CF genotype, such as F508 delta homozygosity, which encompasses a large proportion of our CF population. DOS history, so distal intestinal obstructive syndrome, increases your risk. If you're lucky to live longer than 30 years of age, and as many of our CF patients are now, you have an increased risk for colon cancer. And as I mentioned, post-lung transplantation. And unfortunately, in all of our CF patients that are diagnosed with colon cancer, we're seeing this before the age of 50. And the majority of colon cancer appears to be right-sided. This is courtesy of Steve Freeman from the Galaxy study. So the clinical presentation of GI cancers in CF are difficult because not only are they happening at a younger age, so you don't expect cancer to come to your differential at such a young age, and also delayed diagnosis due to a lot of our CF population has GI symptoms, as has already been discussed. This is the Galaxy data, which shows, a quarter of patients have symptoms that I would say would be mimickers of colon cancer symptoms: incomplete bowel movements, straining, fullness, bloating, and distension in those adult patients. And remember that the most common GI cancer is colon cancer. So there has been some data already in colonoscopy and cystic fibrosis. This is a pivotal study from the University of Minnesota colleagues from 2008 to 2015. They looked at screening colonoscopy at age 40 in CF, so already younger than our general population screening recommendations. And what they found were that in almost half of these patients, you had precancerous polyps, these adenomas, detected on that initial screening, again, younger age screening. 25% had one or more advanced adenomas, so these have a higher risk of colon cancer, and this is 5 times higher than our general population. And a lot of these patients had more than one of these precancerous polyps. So more than 3 increases your risk significantly in the general population. And even if they had a negative initial colonoscopy, if you followed up these patients within 49 months, almost half had these precancerous polyps again, and half of these with advanced adenoma, so that higher risk polyp population. There have been a number of colonoscopy studies in CF, and you can see all over the world: Australia, Minnesota, Canada, and for a number of reasons. So colonoscopy, when it's diagnostic, that would mean you would do a colonoscopy for symptoms versus screening, which is asymptomatic patients. You can see the age of colonoscopy is quite young in the CF population. And so the number of patients is also small in these population studies. But the adenomatous polyp finding and colon cancer finding is really remarkable for the high number of precancerous polyps, as well as colon cancer in this young population. So risk factors for colon cancer, these are the traditional risk factors I think of. Family history, inflammatory bowel disease, environmental factors certainly play a role. We try to say decrease your red meat consumption. But my hope is that you come away from this talk understanding that CF and CF post-transplant is a very high risk for colon cancer. So it's not surprising that in 2018, CF colorectal cancer screening consensus recommendations came out. And those guidelines recommend shared decision between the patient and provider, jointly managed by the CF healthcare professional and an endoscopist. Colonoscopy was the only recommended screening tool for colon cancer. They had insufficient evidence for some of those cooler things like CT colonography, stool-based testing, or FlexSig, which only looks at the left colon. But they did say stool-based FIT testing may be cost-effective and an attractive option, but the characteristics of stool-based testing in CF were lacking. We also know that CF colon preparation is difficult. And actually, I would say it's difficult for even people who don't have CF, okay? So it's an intensive bowel prep. And in CF, we often would recommend 2 gallons of Go Lightly instead of 1 gallon. And Go Lightly does not go lightly for most people. And so you can see on the left side, we really need to see. We can't find little polyps that can be developed into colon cancer. And we really need this really clean colon, but it's very difficult in our CF population. So this leads to our study. Why do we need more research on this? Well, we know that stool-based colon cancer screening is already recommended for the average risk general population. So it may be valuable as a first-line test for colon cancer screening in CF, which is a high-risk population, but it has not been evaluated. We know that the prep is hard, whether you have CF or not, and we know that CF patients are at high risk for colon cancer. But colonoscopy is maybe high risk in CF, right, with pulmonary symptoms and disease. And it's also difficult to schedule. They're already trying to balance seeing so many different providers. So I've highlighted that CF is a GI cancer syndrome. So we initiated the NICE-CF study. We took a long time to think about the right name. So non-invasive colon cancer evaluation in CF. I had the privilege of leading it, but it is a team-based effort. The co-investigators, Steve Friedman and Sarah Jane Schwarzenberg, who's been mentioned a lot in this symposium. Karen Stukowski's here, and her data coordinating center team is based in Seattle. St. Louis University, research coordinator and research nurse, Frida and Sarah, regulatory, and then multiple centers that I will highlight in a minute. It is a team-based approach across the U.S. It's an observational study, and the goal is to determine the test characteristics of each stool-based test, the FIT and the Cologuard, compared to colonoscopy as the gold standard in a cross-sectional study. So our goal was 350 participants to account for drop-off due to missed colonoscopies or inadequate bowel prep, with at least 226 with Cologuard and colonoscopy to compare, and 204 with FIT and colonoscopy. And I'm happy to say that we met enrollment numbers last week. So thank you. Our primary endpoint is to look at any adenomas, so polyps or cancer, so anything that can turn into cancer or as cancer, comparing the negative predictive value, comparing the tests with colonoscopy, looking at sensitivity, specificity, and positive predictive value. We also looked at measures of feasibility and clinical outcomes, such as the prevalence of these precancerous lesions, those advanced adenomas I mentioned, colon cancer, sessile serrated lesions, which are another precancerous polyp. We also wanted to see how many study participants who could actually complete stool-based testing or complete colonoscopy and stool-based testing, and then compare the data between the stool tests and colonoscopy. Today, I'll present the qualitative surveys on preferences and ease of stool sample collection and the colonoscopy prep. These are the 26 participating sites across the U.S. So we have representation from Atrium, Barnes-Jewish, Boston Children's, Columbia, UPenn, Johns Hopkins, Mass General, Medical College of Wisconsin, MUSC, Michigan, National Jewish, Nationwide, UNC. These are the lead PIs as well as the RCs, but we know it's a team effort, and a lot of you guys are part of this team. Oregon, Pittsburgh, St. Louis University, Stanford, UCLA, Kansas, Miami, Cleveland, Minnesota, Southwestern, Utah, and Washington, and Vanderbilt. Again, really appreciate the team effort. So our study design is that patients could be enrolled over the phone or in clinic, and we get basic demographics, give them the stool kits, discuss how to do that. They do an enrollment survey to get their assessment and preferences for colon cancer screening, what they know before they embark on this journey with us. They collect their stool samples. We ask them how it was to collect the stool samples. And then they're all scheduled for screening colonoscopy because they're due. That's the clinical part, not part of our study. So then we collect that within ideally a year, and then post-colonoscopy survey results, and then we compare the colonoscopy data. When we froze the data, I think it was late August, we had 385 consented. Not everyone was enrolled. As you can see, we lost about 43 that were not enrolled. Of those enrolled, 31 did not complete stool kits. And then of the ones that did complete stool assignments, 311 completed them. You can see not everyone was able to successfully complete a Cologuard and a FIT. So that was 262, 77% of enrolled. Some people were only able to give a good Cologuard sample, and some were only able to give a good FIT sample. Of our population, you can see it's a little bit older than some of the earlier colonoscopy studies, but still younger than what we think of for colon cancer screening. But mean age of 47, equally matched in terms of men and women. As with most CF studies, 96% Caucasian. 4% Hispanic, although we really tried to increase that number in terms of enrollment. And you can see pretty uniform distribution across the US, again, due to amazing teams that we worked with. So what we found in some of our preliminary data to date is that most of the people who participated felt that stool sample submission was on the side of easy, one, over being hard, although some people definitely felt like it was hard. In terms of likelihood of submitting annual stool samples, you can see that most, again, felt that they were likely to submit annual stool samples, which you would have to do for FIT. Cologuard is every 3 years, over those that were unlikely to submit. In terms of ease of colonoscopy prep, again, you can see a wide distribution. Again, this data is very preliminary, but some people felt the colonoscopy prep was easy, and you can see a number of people felt it was very difficult. In terms of likelihood to complete a screening colonoscopy, again, you can see that most people were likely to complete a screening colonoscopy every 5 years if that was the only recommendation over those who were unlikely. In terms of preference for colonoscopy or stool testing for colon cancer screening, you can see, even though this is all preliminary data, most people prefer a stool sample as opposed to colonoscopy. So we're still awaiting our primary endpoint, but what we see from this preliminary data is that stool testing is feasible. We don't know how it performs yet in terms of the real outcome measures for precancerous lesions or cancer. But my take-home points are that you remember that CFTR mutations play a role in GI cancer, and to think of CF as a hereditary colon cancer syndrome. And then all patients with CF at this time should be screened for colon cancer with colonoscopy and need extended preps. Some people anecdotally say that they are not requiring as extended of a prep in our CF patients on modulators. And keep in mind that obviously some of our CF patients do feel the prep is difficult, but most would repeat colonoscopy at regular intervals based on our preliminary data. And in terms of stool-based testing as an option in the future for colon cancer, it does seem to be feasible based on qualitative surveys. More people felt stool submission was easy. More were willing to submit stool samples than not. And stool testing seemed to be preferred to colonoscopy. So again, stay tuned for our final results. Like I said, we just met enrollment numbers, so we're doing data cleaning and getting the rest of our data. So we're very excited about that. I think we'll have the final results by next year. And this study, as a reminder, is the largest study to evaluate stool performance characteristics in a population at high risk for colon cancer. And so we will shed light on whether people with CF will have more choices in colon cancer screening, as it does seem like they would prefer stool-based testing over colonoscopy. And I'm happy to answer questions, but remember that we have a poster at 12:15 and 1:15, and our DCC lead, Karen Stakowski, will be there if you have more questions and want to keep talking about colon cancer in adults. Thank you.
So I'll ask one quick question. I guess it's a little bit of a deviation, but we're starting to see CFTR just having one mutation seems to be a risk factor for cancer, breast cancer, colon cancer, everything else like that. So I don't know how much we really come across this in the CF environment, but is there any difference in screening if you know somebody has a CFTR mutation as opposed to someone who does not as a risk factor?
I think right now, if there is some data out there that people with one mutation still have an increased risk. And so I think it's a case-by-case basis. Again, talking to the patient, sometimes if they know there's an increased risk, they will want to do early screening. And I think it's reasonable. I think the guidelines are not clear-cut because there's not good data.
Thank you so much.
Yeah, thank you.
Thank you, everybody, for being here. Appreciate it.